Hello everyone! So the purpose of this video is to describe the regulation of fatty acid metabolism in as abbreviated form as possible, since I know that our time is short at this time of the semester. So this is a nice summary, I think, of both fatty acid degradation and synthesis. So if we look over here on the left, triacylglycerols are broken down in the adipose tissue to fatty acids. The fatty acids leave bound to albumin and enter the bloodstream. And from there, they can go to the liver or to the muscle for degradation. So the beta oxidation pathway is what is involved in breaking down the fatty acids to acetyl-CoA. Every cycle, remember, produces an FADH2 and an NADH. So if you have something like palmitate, which is 16 carbons, you'd go through 7 cycles, with the last cycle producing the 8th acetyl-CoA. So you'd produce 7 FADH2s and 7 NADHs, and you'd get 8 acetyl-CoAs. As long as OAA is available, acetyl-CoA can enter the citric acid cycle to get more NADH. So one turn of the citric acid cycle gives us 3 NADHs, 1 FADH2, and GTP, which is energetically equivalent to GTP. All of the FADH2 and the NADH generated in fatty acid breakdown enters the citric acid cycle, with FADH2 providing 1.5 ATPs and NADH providing 2.5 ATPs. If OAA is not available, then acetyl-CoA forms ketone bodies. And then, and again, that happens in the liver because in the liver we have gluconeogenesis. If OAA is being diverted to gluconeogenesis, it's not available to condense with acetyl-CoA in the citric acid cycle. So the liver synthesizes ketone bodies from the acetyl-CoA. Those leave the bloodstream and can be used by other body tissues such as the muscle and the brain. Acetyl-CoA is also used for cholesterol and other steroid biosynthesis. All right, on the other side, during the fed state, acetyl-CoA is used, it is assembled to form fatty acids. Here we need ATP. We need ATP to convert the acetyl ACP to malonyl ACP, and we also need NADPH as our proton and electron donor in the fatty acid synthase reaction. And the fatty acids can be used to make membrane lipids, or they can be assembled to form triacylglycerols. So this is a nice summary of how all of that is regulated. So what I want to do is I'm going to take this figure and break it down into little pieces and talk about the regulation. So if we're over here, if we are in the liver in the fed state, so in the fed state, insulin will be secreted by the pancreas. And remember that insulin stimulates phosphoprotein phosphatase 1, and that is, as all phosphatases do, catalyzes the hydrolysis of a phosphate group. And here, it hydrolyzes the phosphate group from the acetyl-CoA carboxylase. So acetyl-CoA carboxylase is inactive when it's phosphorylated, but when the phosphate group is removed, it is active. So now acetyl-CoA carboxylase can catalyze the conversion of acetyl-CoA to malonyl-CoA. Then malonyl-CoA can enter the fatty acid synthase pathway to form palmitate. And if you need fatty acids that are either longer or have double bonds, there are enzymes in the endoplasmic reticulum that are able to synthesize those. The fatty acids are then esterified to glycerol to form triacylglycerol, and then they are packaged up into the very low density lipoproteins that leave the liver and can circulate to other body tissues. So here's another diagram of what that looks like. So here we're going to focus on the liver. So in the liver, in the fed state, fatty acids are synthesized to form triacylglycerols, and those are packaged into the VLDLs. Those leave the liver and circulate through the bloodstream, and they're taken up by the peripheral tissues that need them. And as the triacylglycerols are removed, the particles increase in density, so they become intermediate density lipoproteins. And again, as the triacylglycerols are removed, we form low density lipoproteins that are enriched in cholesterol. Peripheral tissues can take those up as well, since cholesterol is the starting material for steroid biosynthesis. Okay, so continuing on to what's happening in the fed state. So here we are um, in the uh, in the adipose tissue. So here's the very low density lipoprotein that's just arrived from the liver. On the outside of the adipocyte, we have an enzyme called lipoprotein lipase. And the lipoprotein lipase will act on the triacylglycerols and interestingly hydrolyze the fatty acids and then resynthesize them. So that's how it happens. The triacylglycerols from the very low density lipoprotein are brought into the adipocyte, hydrolyzed, and then rebuilt. So now we have triacylglycerols. The free fatty acids are used to make the triacylglycerols, and now the excess fat is stored in our adipocytes. So that's what happens in the fed state. So we're storing the products of the food that we've eaten as excess fat, and we can call upon them in situations of fasting or exercise. So again, in the fed state, if we are synthesizing fatty acids, we don't want to be breaking them down at the same time. There isn't any reason to do that because we're fed. So the carnitine transporter that allows fatty acids, the fatty acyl-CoAs, as linked to carnitine, what allows them to go into the mitochondrial matrix for breaking down. We want to make sure we inhibit that process. So high concentrations of malonyl-CoA will inhibit that transporter. So that ensures that the fatty acids stay in the cytosol and are not transported in the mitochondrial matrix because that's where they'd be broken down. So during the fed state, we're going to want to inhibit the breakdown of fatty acids. So how this happens in the fasted state, okay, so let's transition from the fed state to the fasted state. So now we're in the fasted state. So we've eaten, we have a store of fat in our fat tissue. So now what happens when we're either fed or we're exercising? So in the fasted state, we have the hormone epinephrine, so that's shown here as an E. So this cell right here is an adipocyte. So here's the membrane. And the epinephrine binds to a G protein coupled receptor. So the epinephrine binds to its receptor, and the receptor activates the G protein, and the G protein swaps GDP for GTP, and it also jettisons the beta and the gamma subunits, and now we have an activated alpha subunit with GTP bound. That activates the enzyme adenylate cyclase, which takes ATP and synthesizes cyclic AMP. 4 molecules of cyclic AMP will then bind to protein kinase A and activate it. And what we get now is the protein kinase A does what all good kinases do, and it catalyzes the phosphorylation of cellular targets. So in the fasted state, in the adipocyte, what that means is that when the protein kinase A is activated, so in this picture, the entire activation cascade isn't shown, but I did show you that on the last slide. The protein kinase A phosphorylates its targets, and one of those targets is a lipase called hormone-sensitive lipase, and it's called hormone-sensitive lipase because it's sensitive to epinephrine. So hormone-sensitive lipase is phosphorylated, and phosphorylation makes hormone-sensitive lipase active. So that's why it's shown here in green. The hormone-sensitive lipase takes the stored triacylglycerols and hydrolyzes 1 free fatty acid, 2 free fatty acids, and then it needs help with another lipase. And now we have 1, 2, 3 fatty acids and glycerol. So glycerol is water soluble. It has 3 carbons. It can go into the bloodstream and be taken by the liver, and it can undergo gluconeogenesis. The fatty acids can leave, but they are detergent-like, so they have to be bound to albumin in order to be transported through the blood. So what's not shown here is the other enzyme that is going to be inactivated, although that will be in the liver, is the acetyl-CoA carboxylase. So acetyl-CoA carboxylase, when it is phosphorylated, is inactive. So again, here we are in the fasted state. So in this figure, this is the same figure I showed before. So the left half was the fed state. So now let's focus here on the right half. So that's the fasted state. So the hormone-sensitive lipase is now phosphorylated and it is active. So what that means is that the triacylglycerols that are stored are now broken down by the phosphorylated and activated hormone-sensitive lipase to free fatty acids, which then leave the adipocyte and are released into the bloodstream bound to albumin. So again, fatty acids are too nonpolar and too detergent-like to be able to go through the bloodstream on their own. Okay, so now here we are in the liver. So let's look down here at the right and the bottom. So here's the fatty acid albumin complex arriving from the adipocyte. So here's the fatty acid, and it enters the liver, and the fatty acid is converted to the fatty acyl-CoA, and then it has to be transported into the mitochondrion by the carnitine transporter. So that means that we would have swapped the coenzyme A for carnitine, and that it gets swapped back. So now here we are in the mitochondrion. So the fatty acyl-CoA then enters beta oxidation, and the products are acetyl-CoA. And as long as OAA is available, acetyl-CoA can enter the citric acid cycle. But if it's not, because remember we're in the liver now, and the liver carries out gluconeogenesis, if OAA is not available to condense with the acetyl-CoA, then the excess acetyl-CoA forms ketone bodies. The liver makes the ketone bodies but can't use them itself, so the ketone bodies are shipped off to other tissues. So this is a nice fasted state summary. So what's happening here is we have our hormones binding to receptors to activate protein kinase A. So again, over here on the left, this is the adipocyte. So the hormone-sensitive lipase becomes phosphorylated and active, and these are the lipid droplets that are used to store the triacylglycerol. So there's a hydrolysis of these three fatty acids from the triacylglycerol. Now we have the free fatty acids, and we will take them and bind them to albumin, and we'll send them to the bloodstream. And then here, the last slide I showed the fatty acids going into the liver, but they can go into the muscle too. So muscle can use fatty acids as a source of energy. In fact, heart muscle actually really likes fatty acids. So this is a myocyte or a muscle cell. So here, there's a fatty acid transporter. The fatty acids come in, and again, they go through beta oxidation. And here you don't have a worry with OAA being diverted because muscles don't undergo gluconeogenesis. So the fatty acid is broken down to acetyl-CoA, and it goes through the citric acid cycle and then electron transport, and you get lots and lots and lots of ATP to power the muscle when you are fasting or exercising. So that is the summary for the regulation, and here are my references. I hope this is useful, and I will see you next time.